Hello, everyone. So we are now on to video number 4 in this series, and we will all get to experience the beauty and the power of cluster analysis for the diagnosis of hematolymphoid neoplasia. Now, in the next video, chapter 5, I will acknowledge my own biases about this, but right now is the time for me to get onto the soapbox and try to convince you that this is the best way to approach clinical flow cytometry data. So let's get to it. At the end, we are going to do some cases, so where I will analyze them in front of you. And maybe we'll leave with a case as a quiz, which you can then answer either down here in YouTube, or if you found this to Twitter, then you can answer the quiz in Twitter. So here we go. So as I told you in the previous video, cluster analysis is the approach that we use to evaluate flow data. And in this type of approach, which is distinct from gating or sequential gating of populations, the one difference is that you're looking at all the data at all times. And this is just an example of a 6-parameter or 4-color flow that I gave you before, where you would keep looking at these populations at all times, whether it be blasts or granulocytes or monocytes. You wouldn't make assumptions of where to draw the gate. You would let the population tell you where to draw it, and you would use all the parameters to hone in on a blast population or a monocyte population. And this will become more clear when we go over real cases. So before we go there, this is a case that was developed by my dear friend Steve Croft. We were both at UT Southwestern together. We drank the Kool-Aid of cluster analysis that was brought to that institution by Louis Picker a long time ago. So a quick case, here's a bone marrow specimen that has come in for an evaluation of lymphoma. Is it a stage 4 lymphoma? Is bone marrow involved? Basically, that's the question. And so instead of having all these 15 different things open in front of you, we have honed it down here to 3 plots. So this was 4-color analysis back then. And you have your forward scatter and side scatter, so size and complexity. You have your CD20 and CD38 in this tube, and you have your Kappa and your Lambda. And the way you approach it is, it's good to start with the lineage specific marker. So on the screen, what I would do is I would go ahead, take the blue color, let's say whatever color you want, blue color, and I would paint all these cells as potential B cells blue. And that would look something like this. And as soon as I paint this based on these two parameters, instantly the software knows, or the file knows, what other parameters are saved on these same events, and so it would light up. And now if you go to the forward scatter versus side scatter, you can see that yes, B cells should be in this lymphocyte cluster right here, and the rest is just junk or scattering everywhere. So you would use these 2 dimensions to hone in on this cluster. And what will happen when you clean all this up, and we'll do this in the cases so you know what cleaning up looks like. Obviously, this stuff that is way on the ceiling over here would also disappear. They're probably just junk that is autofluorescing and stuff like that. So we would clean that up. And as soon as we cleaned up this based on this cluster, all this stuff disappeared. So that looks nice. And now we would try to do a Kappa Lambda ratio on these B cells to see if they're polytypic or if there's any monotypia involved. They look like they're splitting on 2 sides. For Kappa Lambda, as I've told you before, you go down this diagonal and you can override this blue color to make it some other color. And that's what was done here. So there is a pink or violet population, violet population and a population on the Lambda side. And turns out that the Kappa Lambda ratio was calculating out to 0.7 to 1. Now, in most approaches, here is where we would end. We wouldn't be sure what is going on. I've told you that the normal Kappa Lambda ratio should be between 1 and 2. So 0.7 is actually a low ratio, which means if there is a tumor in here, it would be lambda, right? It is driving the kappa to lambda ratio down. So the tumor should be blue in color if there is any tumor here. Otherwise, this may just be something that we couldn't figure out. But if you look just right, seems like the blue color is preferentially there in this slightly larger part of this size spectrum. Even here, seems like the blue color is more into the CD20 bright. Let's take a closer look here, right? The blue is bleeding more into this larger than the violet is. Same way the blue seems to be brighter. So what we could do is hone in on the population in some different way. So in this particular case, Steve ended up getting an extra marker with CD20 and CD22 included. And so in this new tube, as you see, there is CD20, CD22. And you can see that on the CD20 bright side, where we were expecting a population, CD22 is also bright. So if you squint just right, maybe this is a cluster that you could divide out of the B cells. So here are B cells, here are some more B cells. And when you paint this based on these two markers and now look at kappa lambda, turns out all those CD20 bright events also very neatly lie on the lambda side of this diagonal. And then the kappa lambda ratio amongst the blue events becomes normal. And clearly now you can be 100% confident that there is a lambda restricted population in here. The peripheral lymph node would corroborate whether that was lambda positive. That would make this case positive. And it is a very strong evidence that the bone marrow is involved. So this type of honing in on populations and looking at clusters is what this analysis gives you the power to do. To me, again, to get on my soapbox, this is much more like a pathologist's approach to looking at flow data, similar to how when you're sitting at a microscope, you want to scan it at low power in the way you like. You want to determine where you want to go and look at high power. You want to determine what markers to get. And this is sort of that kind of approach where all data is available to you. Somebody else has not gated and taken pictures and sent them to you, and so on and so forth. Now, many different softwares can get you there, and people are trying to automate this more and more, and we'll see where the field goes. But at least right now, this is the way that I feel you will provide the best education to the next generation of pathologists if they approach flow this way, where they are themselves playing with the data and not relying on somebody else's expertise to have pre-gated and drawn gates, so on and so forth. So with that kind of case introduction, let's go to the actual software and see how this works live. Just a reminder of where populations will fall. And this algorithm is what we would use in a couple of the cases. So if you want, you can pause and revise this. I went over this in a previous video. And then we will now switch over. I will share a different screen. And this is just what I will do in the next video, where we will discuss the story behind this hashtag, go with the flow. So I am about to share a different screen. Okay, so we are in this software now called CytoPaint, and I have opened my template here. We used to have a software called Paint-A-Gate, and now the latest one that can do lots of colors is called CytoPaint. And what I'm going to do is I'm going to load a file here, right? And you can see this is a B cell tube. I see CD19 here. It's got CD45. This is usually the favorite dot plot of flow cytometry labs, where all the gating begins here. I will suggest we should do it a different way. Here's forward scatter versus side scatter. You see the lymphocyte cluster. These are your monocytes. This is granulocyte, seems like a blood specimen, and it has come in for a lymphoma evaluation. So the way we do it is we can pick any color. So let's say I pick color red, and So there are three primary colors here, red, green, and blue. Then yellow, violet, and cyan are combinations of two of those, and all three together make white. White overrides everything. So here's red. And let's say what I find helpful is to start with a lineage-specific marker so that you know what you're looking at, and you're not assuming that some gate drawn here is equal to a blast or a lymphocyte or whatever. So this was a B cell tube. CD19 seems to have a very nice cluster here of positive cells. So the first gate, you draw it broad, and then you hone in on the population. So I'm just going to say, okay, what about all of these? Let the cluster tell you, okay, these are potential B cells. And now you can look at this forward scatter versus side scatter, and you can see a very nice cluster here of lymphocytes. So now you go to white color, and you go in and clean up everything that you think is not belonging to this cluster. I tend to leave the large cell in there in case there is a large cell lymphoma. Eventually, once you convince yourself this is not something weird, you could get rid of it and stuff like that. So here's your lymphocyte population. Now you can use your 45 versus side scatter. Nothing against CD45. And again, use this to hone in on the cluster that you're interested in. Okay, so this seems like a reasonable B cell cluster. And now we look at the Kappa Lambda, okay? So here's your Kappa Lambda. I'll just blow it up for the purposes of this talk. And what can you see? You can see that there is a cluster of Kappa positive cells here. There is a cluster of Lambda positive cells here. These look like maybe your polytypic B lymphocytes in the specimen. And there is clearly a barren cluster here that is on the left side of the diagonal. So it's Kappa, and so it is restricted to Kappa and is quite dim. So just for us to be sure, if you did a Kappa Lambda ratio on this whole population, so you can take a different primary color and overwrite one side. So whatever was red turned violet, and then you can just subtract the blue population away, and I'm going to subtract the white population away as well. So you see, you saw this overall Kappa to Lambda ratio very, very high. So see, this is 51% down to 0.74. But what about if you just hone in on this one? So now I could take a third primary color just for fun and override this population with that. It would become white because all three colors are there. And now the Kappa to Lambda ratio is 0.9 to 0.7. So Kappa Lambda is now normal in these cells. So I'm going to merge that white color to something primary, let's say green, just so you guys can see it, okay? So you got that. These are your polytypic B cells in the background. They are behaving themselves. So what I'm going to do is now I'm just going to hone in on this violet population just for the purposes of this demonstration. So, okay. So this is our population of interest. I'm going to take away the white, and I'm going to make violet red. Okay, so we are back to our population. And now we are sure that this is a monoclonal B cell population, small cells, right? Predominantly small cells. And so what are the two markers we're going to look at? We're going to look at CD5, which is right here, and CD10. So seems like the population is CD10 negative. What do you think these are? So these are your granulocytes, by the way. Granulocytes are CD10 positive. So just right there, they light up. So you can undo that. And this population is also, it's CD10 negative, but it is CD5 positive. So regular B cells would lie right here. This clearly stretching out into the CD5 positive zone, almost as bright, some of them, as the mean brightness of these T cells. Just so you know, these are T cells, see nice lymphocyte cluster, CD5 positive. T cells tend to be slightly brighter for CD45 than B cells. I'm just sort of rattling off things randomly as they come to my mind. But here, so now you have a CD5 positive population that is Kappa restricted. And so what's your differential? It's either a CLL, SLL, right, or a mantle cell lymphoma. What do you look for to resolve that? Well, one thing you can look for is CD23 versus FMC7. There's a fair amount of autofluorescence here on this channel, but we know by experience that this is FMC7 negative and clearly CD23 positive, okay? So it's a CD23 positive, FMC7 negative. And the other thing, of course, we look at is right here. It's a little bit down in this template, is your CD200. And clearly, your population is CD200 positive. So you have a diagnosis now, right? So this is a CD5 positive B lineage non-Hodgkin lymphoma, and it has the immunophenotype of a CLL, SLL. Then, of course, you go back and do the counts, make sure this is not some monoclonal B lymphocyte. Clearly, this patient has a CLL, SLL, all right? So that's how you go about doing this type of analysis. Let's go do one more case. So I'm going to load a different case here. Let's try this one. This tube. Okay, so here's a bone marrow. And again, let's say this has come in for evaluation of a lymphoma. So again, you can pick any color, but we're gonna start, it's a B cell tube again. See, it's got CD19, but there is also T cell. It's sort of a screening type tube where B and T is combined together. But let's look at these B cells in detail. So you could, again, CD19, then, so it's like playing a video game, honing in on populations. The amount you will learn about these markers, even on reactive and normal populations, is a lot more when you handle the data yourself like this. And here's a great example of why gating strategies might sometimes take you out of the interesting population. So here's your B cells, and clearly there are almost 3 clusters of B cells, right? And they go pretty dim on CD45. And you probably know what is going on here if you've done flow cytometry for some time. But here are your 3 B cell clusters on CD45 going pretty dim. So if you had just gated on the bright CD45s, you would have missed part of these, right? So in fact, let me show you. If you had gated only on the top one initially, then you would have missed a bigger chunk of them. And of course, if you had gated up to here, you would have missed these. And so many labs, of course, gate down to the dim one, but then you're conflicting with quote unquote the blast gate. So what is this CD45 dim population? So it seems to be small and large. I'm going to get rid of the white here. So you're focused in. All CD19 positive. And almost all of them are CD10 positive. Surface immunoglobulin predominantly negative and streaming into Kappa and Lambda. My residents here at Iowa would know exactly what this population is because it's one of my favorite cell types. Once you train at UT Southwestern, you cannot not like these cells. So clearly this CD10, so I went in and painted all the CD10 positive cells together so you can get an idea of the entire population. And you can see that it's mostly surface immunoglobulin negative, but streaming into Kappa, Lambda, these are basically hematogones, right? These are developing B cells in the bone marrow, going to B cell school as they grow and mature into these blue mature B lymphocytes. Another thing you could have done if you wanted to do it a different way is you could have said, okay, right from the B cell population, I'm going to do a Kappa and Lambda ratio. So I'm going to circle the Kappa to make it violet. Then I'm going to circle the Lambda, all of it, to make it cyan or light blue. And now I do a violet to cyan ratio, so about 5:3. So it's a normal polytypic B lymphocytes over here. So the B cells are normal, except there's a lot of hematogone hyperplasia. 16%, even if you don't include the streaming ones, really you should include all of them. So this is a case that has hematogone hyperplasia, and you would go back and see if that made sense. Is this a regenerating marrow? Is this some kind of ITP, autoimmune disease, you know, other causes? So it's a cool case of lots of hematogones. Now let's just do one more case and we'll end this one, and I'll sort of end it as a quiz so that you can then tell me what it is. So here's a case for you, and it'll also show one more aspect of this cluster analysis pathway. So here's again CD19, just like we did in the other. I'm marking these, and then I'm going to use forward and side scatter to clean it up. Now clearly all this up here is junk. Normally, you know, one would think of this as the lymphocyte cluster, but there's a clear cluster here. And so before you do something like this, either when you're gating or when you're analyzing it this way, watch out, right? So you definitely want to make sure you're not taking away something that may be of great interest. And the way to do that is you could overlay it with a different color, like green in this case. Take away the green, take away the white, and now you have this population that you could have gotten rid of. See, even in the CD45, if you did a lymphocyte gate, the usual gate, you would not be including these B cells. These are B cells. These are 19 positive. Here, where are they? These are 19 and CD20 positive, right? Nice and bright for 19 and 20 right here. So you would have not even considered this population in the beginning. Maybe you would have gated it as a monocyte gate and then found it, which is actually turns out to be a big clue in this case. So you got a CD19, CD20 population, and look what's happening here. So let me just hone in on these bright ones. They're clearly lambda restricted, right, on this side of the diagonal, dimmer than the normal B cells. Now, if I wanted to see what the other cells are doing, I could do this, and I would get a kappa, right, to lambda. So violet to blue ratio of 1 to 0.88. So this is a 0.88 on the lambda side. So this ratio is normal. So the main population to be interested in turns out to be actually this cyan population. So I can hone in on this, and this happens to scatter in such a funny way, and it is in such a funny location, right? But it is a monoclonal population that seems to be FMC7 positive, 23 negative, but what do you look at next? You look at 5 and 10, right? You've got a monoclonal population. You look at 5, seems to be negative for 5. You look at 10, seems to be negative for 10. I haven't cleaned this up very nicely because we are rushing through the case. But here's a 5 negative, 10 negative population. So none of the other markers matter as much in this scenario. I mean, yes, it is CD200 positive, but it's not a CD5 positive in the first place. So 5 negative, 10 negative, what's your differential? A marginal zone lymphoma, a lymphoplasmacytic lymphoma, and a hairy cell leukemia. And there's something about, you know, not having monocytes here, the weird scatter of these cells. So you go to the next tube where we have some other markers to help us out. And we'll try to find these. So here's your CD19 in this tube. Again, go for it this way. And this time I will sort of rush through it because we know they see the same scatter here. So why not just go in and hone in on this population? See, this fun you cannot have, drawing all these curved lines and painting through this like an artist. you cannot have that in a gating thing where you're clicking all these acute angles. Anyway, enough of that. So now this is a 19 positive population that, let me get rid of the white here. And while you think all these clicks and everything is less efficient and might be slow, it's really not once you get used to it. This can go much faster than the back and forth that happens in regating and reevaluating. So CD20 bright, so I'm going to hone in on that cluster. And now I'll let you look at these markers. Look at these 4 plots. All the axes you can pause if you want, and then let me know the diagnosis. Fairly easy. So we will stop here for today. In my next video, I'll talk about the general story of Go With The Flow and maybe end with another case or 2 as quizzes. And then I think we are ready to jump into cases on a regular basis. And we'll continue this kind of cool live analysis of exciting cases as we go along. So I will stop the share here. Thank you for your time and see you in the next video.